Hi guys, welcome back. I'm Mr. Khor, and this is the second video for Chapter 5, Form 4, Chapter 5, Metabolism and Enzymes. Now, the earlier lesson, the earlier online video, we have discussed about all the following, definitions of enzyme, why we need them, the naming, remember the two types, one with ASE, one without. Characteristics such as specific in action, they are sensitive to temperature as well as pH. Anyhow, I'll still be going through this again. Okay, sensitive to pH and temperature. Alright, what else? They are not destroyed. Okay, tidak dimusnahkan. They are not destroyed, not altered, meaning not changed after a chemical reaction. So all those we have seen in the previous lesson. Now, moving on, this next area that you're looking at here is actually a very important component. Now, for the old syllabus, KBSM, they love to ask, explain the formation of extracellular enzyme. Menghuraikan pembentukan enzim luar sel. Okay, huraikan pembentukan enzim luar sel. Okay, first of all, okay, these are the two types of enzyme now. What is the difference? What is the main difference between these two? Check. Intracellular enzyme, let's say they are produced in the cell. Okay, for example, let's say this is a lysosome. Okay, inside got enzymes. Now, these enzymes are known as intracellular. Intracellular means they are produced and function within the cell. Okay, dihasilkan dalam sel, berfungsi juga dalam sel. Okay. While extracellular, they are produced in the cell, dihasilkan dalam sel tetapi dihantar keluar. It is, we use the word, secreted out of the cell in order to function. Secreted out of the cell to function. So the main difference is intracellular enzymes, they function in the cell, extra function outside the cell. Okay. So, fellow students, this is what you're required to memorize for essay, for essay purposes. You can draw 3 stars. You can draw 3 stars for this particular process. Okay, in my notes. Then we'll go through. Alright, let's start, guys. Let's say exam asks you production of extracellular enzymes, let's say 6 marks. Now, let's say essay asks you 6 marks, means you have to come up with 6 points. So what is your first point, second point, third point, so on and so forth. Okay, you must be able to answer. Basically, if you can answer them now, confirm you can answer them in the exam hall. Okay, so why? We store all the information in our brain. Okay, point number one, proteins are synthesized by ribosome. Protein dihasilkan ribosome. Then, protein enters rough endoplasmic reticulum. Dalam BM, jalinan endoplasma kasar. Okay. So first point, ribosomes synthesize protein and protein enters the rough ER. Rough ER will pack the protein into a transport vesicle. Okay, vesicle pengangkut and this transport vesicle will carry the protein to the Golgi apparatus. Okay, transport vesicle carry protein to the Golgi apparatus. In the Golgi apparatus, protein is modified, diubah suai. Protein is modified. Can you see the change in color? So protein become enzyme. Okay, protein is modified to become enzymes. Then, now enzymes are packed into a vesicle, but not transport vesicle anymore. Very important. We call this a secretory vesicle. Dalam BM, vesicle rembesan. Kenapa vesicle rembesan? Sebab mereka akan dirembes keluar daripada sel. Okay, for the English one, so after a secretory vesicle is formed, you must mention one more point. I like to put it this way lah. So here you have to say the secretory vesicle. You use an F word, a 4-letter F word. What is the 4-letter F word? F-U-S-E, fuse. Bercantum. It will fuse with the plasma membrane. Secretory vesicle fuse with the plasma membrane and release enzymes out of the cell. Yeah guys, we call these the extracellular enzymes. Okay? Alright. So I'll go through one more round and you'll realize that we're able to settle the whole process in 30 seconds. Okay? 30 seconds. So this is what you need to know for exam purposes. But if you're using visual, visual meaning diagram, pictures, videos, you are able to revise much more efficiently as well as you revise it quicker. Okay, let's start. First point, ribosomes synthesize protein. Protein enters rough ER and is packed into a transport vesicle. Okay, transport vesicle. Transport vesicle will carry protein to the Golgi apparatus. Okay, guys, what happens here? It is modified. Protein is modified to become enzymes. Enzymes are now packed into a secretory vesicle. Okay, F word, F-U-S-E. The secretory vesicle will fuse with the plasma membrane to release enzymes out of the cell. Okay, how long did we take? Maybe 1 minute. But this is only our second time. The moment you repeat the third time, the fourth time, the fifth time, I have students doing it in 10 seconds, guys. I have students doing it in 10 seconds. Okay. Now, two things to touch up on this particular process. Two things to touch up. Yang pertama sekali, if they want intracellular production, intracellular enzyme, the way to explain is, all the points are the same, except the last point. It is not secreted out. It is not secreted out. Okay. That's the first thing. So if they want extracellular, we say enzymes are released out. Kalau tidak, just say it remains and function inside the cell. Okay. Yang kedua, second thing, regardless whether intra or extracellular enzymes, let's say they ask a heavy 8 marks. Heavy 8 marks means you need about 8 points, or better still more. So the two extra points which you can talk about is number one, mitochondria, okay, produces energy in the form of ATP for the process to occur. Mitochondria produces energy in the form of ATP for the process to occur. Number two, Nucleus contains DNA. Okay, let me just write the full sentence. Nucleus contains DNA which stores genetic information for enzyme or protein synthesis. Meaning to say that in order to produce protein in the first place, okay, listen carefully, in order to produce protein in the very first place, genetic information is needed, okay, by the ribosome, and this genetic information is stored in the DNA, which is found in the nucleus, okay. So if it is a very heavy 8-mark essay question, follow back whatever I explained and add on these 2 points, alright, okay. Okay, if you have my notes, this would be page 17, the English version. Now, moving on to the next area of enzyme. Guys, we are required to know the log and key hypothesis. It is a very simple 4-point hypothesis. Very simple 4-point hypothesis. Okay, kita panggil ini hipotesis kunci dan mangga. Okay, kunci dan mangga. Okay, just to do a very quick recap, let's say this is my enzyme. This is called the active site, tapak aktif. Maltose is considered the substrate. Okay, bahan substrate. Substrate always enters the active site. Substrate always enters the active site. Reaction takes place, and then products are formed. Okay, so before the reaction takes place, we call it a substrate. After the reaction has completed, we call them products. Alright? So now, for log and key hypothesis, again there are 4 points. Okay, point number 1. The substrate is represented by the key. Okay, substrate is represented by the key. Or you can see key represents the substrate, while the log represents the enzyme. Log represents enzyme. Second point, the active site is complementary. The active site of enzyme is complementary to a substrate. Complementary means can fit. Complementary means can fit only one type of specific substrate. Okay, for instance, this active site of sucrase is meant for sucrose to enter. Okay, it's not meant for any other substances such as maltose. No, it is only meant for sucrose to enter. Alright. Now, guys, the moment a substrate enters the enzyme, we call this whole thing an enzyme substrate complex. We call this entire thing an enzyme substrate complex. Dalam BM complex enzyme substrate. Okay, dalam BM complex enzyme substrate. Reaction takes place, and now products are formed. Okay. If they want one more point, this is a very crucial point, guys. Very crucial point. We always end with the structure of active site, or you can just say enzyme can be reused again, or enzyme is not destroyed at the end of the reaction. Okay. Alright, I'll repeat one more time just in case you can't catch up just now. Okay, one more time. Okay, first point. Key represents substrate. Log represents enzyme. Second point, active site of enzyme is complementary to one type of substrate only. Or if you do not want, you can say enzyme of, the reaction of enzyme is specific. Now, when the substrate enters the active site, we call this, what do we call it? An enzyme substrate complex. This will earn you 1 mark in exam itself. Meaning in essay, the moment you write this, you should be awarded 1 mark right away. Reaction takes place, products are formed, and guys, what is your last point? Enzyme is not destroyed, or it can be reused again. Okay, enzyme is not destroyed after reaction, or it can be reused again. They are essentially the same point, not two separate points. Maksudnya kalau kamu tuliskan kedua-dua ini, kamu hanya dapat maksima 1 markah sahaja. Only maximum 1 mark. Okay.